Okay, so chapter 10. This is the one that'll take us to our next exam. So what we're doing here is we're putting a few things together that we started out with before. We talked a little bit about what a nucleic acid is, and we certainly talked about chromosomes just now. And so the question is, what exactly does the DNA do for us? I mean, how does it actually produce anything tangible? And so the first thing to do in this part is to talk a little bit about the structure, review the structure, and then talk a little bit about the history of its discovery. Now, there are information storage molecules in us. We identified those before as the nucleic acids. And the two molecules that we refer to by that name are DNA and RNA. And DNA is short for deoxyribonucleic acid, and RNA for ribonucleic acid. And you may ask why are they called that? And so here's your explanation. First of all, the term nucleic acid in both cases simply refers to the type of molecule they are. And if you recall, we had proteins, we had polysaccharides, sugars, we had lipids. Well, this is the fourth type of molecule, so the nucleic acid. Secondly, the phrase ribo in both of these cases, ribo, actually refers to something called a ribose sugar. So both of these have a ribose sugar of some sort as part of it. And if you look at DNA, this particular ribose sugar is deoxy. And deoxy simply means missing an oxygen. Missing an oxygen. So if you translated this long phrase, deoxyribonucleic acid, then DNA is the nucleic acid whose ribose sugar misses an oxygen. This is how you name things in chemistry. And then RNA, of course, is the nucleic acid that has ribose as its sugar. That's much more straightforward. And of course, the reason why I made these bold is because DNA, that's where the abbreviation comes from. Both of these consists of monomers called nucleotides. And if you remember, we had monomers of proteins, those were amino acids, and we had monomers of sugars, those were monosaccharides. And so here again, we have this idea of monomers. The nucleotides have three components: the sugar, and as I just told you, it's some type of ribose sugar, a phosphate group, which is something we saw already when we talked about ATP, and a nitrogenous base, and that's kind of new, so we'll talk about those in a minute. The sugar and the phosphate group, those are the so-called sugar-phosphate backbone, and they form the support structure for this nitrogenous base. Here's what that looks like. On the right, there is a nucleotide. This particular nucleotide is the one that includes as a nitrogenous base thymine. Now here is that nitrogenous base, and the nitrogenous base comes in four different types, and these are for short A, G, C, or T in DNA. Then you see here, there is the deoxyribose sugar, and here's the phosphate group. And where they link is up here, so that goes to the next nucleotide up there, and down here, so that goes to the next nucleotide down there. And if you keep connecting from sugar to phosphate to sugar to phosphate, then you end up with this: sugar to phosphate, or in an abbreviated version, sugar to phosphate, and so on and so forth. And so the thing that always sticks out is that nitrogenous base. And as you can see here, the way they are put together, the A and the C and the T and the G, they all have different structures in some way, shape, or form. So we're going to investigate a little bit more about what that means in a minute. But this is the basic structure of the nucleotide and how a DNA molecule is put together. When it comes to the nucleotides, there are actually 5 types of nucleotides in these data storage molecules. Now, if you remember, we had 20 different types of amino acids, and then we had a few of the monosaccharides. In this case, we have 5 of these monomers. They differ in their nitrogenous base, and so when you look at both DNA and RNA, they have 3 of these in common. The cytosine, the adenine, and the guanine are occurring both in RNA and DNA. And then there's one that's different, whereas DNA has the thymine, that's T, RNA has uracil. So if you write down their complement of bases, what you can do is say in DNA we have A, C, G, and T, whereas in RNA we have A, C, G, and U. So whenever you might see a T in RNA, you find a U. Now, just to give you an example for why this is important, if I give you a sequence of nucleotides, and this is the sequence, is this from DNA or RNA? It's from DNA, because if it has a T in it, it's got to be DNA. That's how you can recognize it. Now, when the DNA structure was discovered, this was a time when we didn't have modern technology, and so some of this work was done with a lot of deduction. It was really like what sleuths would do, Sherlock Holmes, that kind of thing. And in fact, people for the longest time were wondering which of the molecules they observed inside of cells actually were the ones that carry forward genetic information. Of course, they had understood that with Mendelian genetics, that information from one generation would be transferred to the next generation, and one of the candidates for this was proteins. The other candidate was these nucleic acids, and people really didn't know in the early part of the 1900s what those would be for. Eventually, it turned out that there were quite a few people who realized that proteins couldn't contain information because they were doing so many things, so perhaps it was this molecule. And then the question arose, well, if it is this molecule, what is it about this molecule that allows it to store and transfer information? So one of the things they needed to figure out was the structure. And in the late 1940s and early 50s, they started to do a variety of experiments trying to determine the structure and looking at the molecule itself, which of course is a very small type of thing to look at. And the process involved a technique called X-ray crystallography. And the work was carried out by Dr. Rosalind Franklin. And Dr. Franklin worked in the lab of Maurice Wilkins. And so Wilkins and Franklin were involved in the X-ray crystallography part. And this image that you see here, that is an actual image or an actual replication of Dr. Franklin's X-ray crystallograph. And this is something they published. And after they had published, or right as they were publishing it, Watson and Crick figured out the structure of what DNA must be like based on this image. Now, if you look at the DNA at this image from the top down, this is how that is working out. It's a top-down image, and based on a top-down image, you can see that the DNA, if you look at it, it's kind of a rounded structure. And if you look at this image here, this is where they built a model of the DNA, and this is why it's a rounded structure. When you cut it here and you look up or down into it, then this is probably what you see. Now, it's not so easy to make these deductions, and because it was a real leap in terms of knowledge, Watson, Crick, and Wilkins received the Nobel Prize in Medicine in 1962. The paper that Watson and Crick published in a journal was only a page long. I mean, it's not a long paper at all, but it certainly had a huge impact. The reason why Rosalind Franklin was not part of this Nobel Prize is because Nobel Prizes are not awarded posthumously, and she had died of ovarian cancer at age 38. So she was unable to be named as part of the prize. But she and her work are certainly highly regarded and well recognized. So that's how the DNA structure became known. And when people started to try and explain how this worked, they realized that a really good way to do this is to take a rope ladder and twist it into a spiral. So the structure here, this is basically what the DNA would look like. And if you look at the rope ladder and you look at the rope itself, well, the rope, that's the sugar-phosphate backbone. And it happens to be a double helix, so that means this sugar-phosphate backbone occurs on both sides of this rope ladder. And then the rungs are made up of a combination of these nitrogenous bases. And there are different ways by which you can put these rungs together, but clearly if you're going to have a functioning rope ladder, then you have to make sure that the rungs are the same length. And so it appeared then that there must be some rules when you're building this DNA type rope ladder. There must be rules for how you can form these rungs. And this is indeed what happened. And here is how some of this works. If you look at this twisted rope ladder here, you can see that on the one side, right here, there is the sugar phosphate backbone. And I'm going to draw a little arrow here in order to illustrate the way the oxygen is pointed. And then I'm going to do the same thing over here. I'm going to draw that arrow again, but I want to point out now that the arrow goes the other way because the oxygen is pointed in the opposite direction. And so you have one strand that goes that way and the other strand that goes the other way. So DNA is made up of two strands with different orientation, which doesn't really matter. I mean, if you think about a rope ladder, the rope on the rope ladder is produced, if this is the rope's produced in this direction, maybe from A to B. And when you're looking at the rope ladder, that doesn't really matter. I mean, it doesn't matter to you whether the rope is produced from A to B or from B to A. It doesn't matter which way it hangs either. The issue is production. So when this is produced, it's produced in a different direction than that one. That's the only thing to be aware of. Now, as far as the rungs are concerned, if you look at the rings involved, there's one ring, there's a second ring, and there's a third ring. One ring, two rings, three rings. So whenever you look at these rungs of that rope ladder, if you will, there are always three rings, right? 1, 2, 3 rings. And then there's some hydrogen bonding. And it turns out when you look at how this pairing works, there are strict rules. You find that the A always pairs with the T because only there can you make the right kinds of hydrogen bonds that would actually attract and hold together the individual strands on both sides of the DNA. You could not take the G and the T because even though that would be the right number of these the right number of ring structures, then you wouldn't get the right hydrogen bonds and they would not work together as a rope ladder. So whenever you look at DNA and you see an A on the one side of the DNA, then on the other side of the DNA you're going to see a T. That's how the DNA is put together. Whenever you see a C on the one side, then on the other side you're going to see a G. So there is these pairing rules.